we'll talk about those obviously when we redo the citric acid cycle and add to it, right? And moving on from there. Rough endoplasmic reticulum. Those little dots are ribosomes. That's how we make proteins. The ribosomes receive the message in the form of mRNA. They help to assemble the proteins. The proteins get dumped into the rough endoplasmic reticulum as the primary sequence gets produced. We're going to talk about all that in 2 weeks. The Golgi apparatus, its job is to fold, process, and if the protein is being exported, package it so that it can be left outside the cell, or if you're making a lipid, so that the lipid can be packaged and exported. Okay? The smooth endoplasmic reticulum, it makes things, just not proteins. If you don't have a ribosome attached, you're not making a protein. It's smooth versus rough, because if you look under the microscope, the endoplasmic reticulum with ribosomes attached looks rough. It's got little dots on it. But smooth doesn't have the dots. That's the difference between them, and it's because one is producing proteins and the other one is not. Lipids, or if you've got another weird little hybrid molecule. Desmosomes and tight junctions are how the cells are glued together. The nucleus is where the genetic material is held. The nucleolus is where the genetic material to do specific things, including maintain the nucleus, is held. Okay, again, transcription and translation, we'll talk more about those two things. The vacuole, those are storage for lipids, protein, liquids, what have you. They also, vacuoles can be moving from Golgi apparatus to exit, right? Or they can be moving, they can take a bunch of things from here and move them down there. So you can look at it and you don't necessarily know whether it's a transport vacuole or a storage vacuole just based by looking on it. You would have to do a molecular test to see what was in it and know what the cell was doing. You'll just see these spaces when you look at the cell and you'll be like, oh, that's a vacuole of something. Okay, what are the lysosome? This we will, again, this is probably more in B, but this does denigrating of wastes, and this is where we start talking about antioxidants and reactive oxygen species. We have to stop harmful chemical reactions from breaking our cells down. Anything else that I didn't talk about? Our brush border membrane. Our brush border membrane are our microvilli. It's where all these little proteins to interface with the lumen of the small intestine where the food is being held are going to happen. I drew a little wave in here. That's the glycocalyx. It's the thin water layer, and we'll talk about that next week more when we start putting these things in there. The basolateral cell membrane is out. This is to the rest of the body. This is where you've got your lymphatic system, or your lipid transport, or your blood, so you can dump the nutrients in and it can go get processed and be sent where it needs to go. So we're not learning this alone. I'm not quizzing you just on an enterocyte because it's cell bio. I'm quizzing you on it because we're going to put everything that we do into one of these organelles or one of these molecules, 5 of these places. You're going to watch what happens when you take a protein in, where it goes, how it gets broken down, how it gets exported. So we just want to know where that's happening and how. Okay, so I'll leave this up here too, and then we're going to go to see if there's anything else.